Welcome to our next video in the course Bioimage Analysis. So here we're looking at the introduction to three areas. So first is virus particle image analysis, co-localization of fluorophores, and then also measuring label intensities, where we'll do the actually coding part in the demo videos in the other part. So our steps for this whole area is similar to what you might have used if you use different tools. So looking at image import, you've generated some images. We want to load them into our software tool, and that's what we'll do with R. We'll load them in there. We will calibrate the image. So we need to consider if we need to correct for distortions, normalize intensities, adjust for uneven illumination, and so on. We may need to reduce the noise, and then also consider for multiple images, like multi-channel ones or transmast ones, think about how we're linking them together. So this is all part of image pre-processing. Then we perform things like segmentation or thresholding to remove the kind of background intensity that might eliminate our signal. We may do partial segmentation in certain regions, and then also think about manual correction for segmentation errors. We can then extract different features from the segmented images, such as size, shape, number of different particles or things like that. We can identify how many of the objects of interest are present. And then also we can track movement if we have multiple images. We can use this using R, and that will allow us to then to interpret the output. So we'll look at images like this on our right, where we're looking at the number of virus particles or virus factories that are present, and then also the numbers of host cells on two different linked images. In terms of looking at the co-localization of fluorophores, essentially what we're looking at co-localization is the spatial correlation of two different fluorophores in the same sample. So maybe that might be the red, green, and magenta overlap. And we have these two different fluorescent labels, and we're identifying, do they co-occur, and therefore is there some sort of interaction by the associated proteins. So we get the images with those fluorophores, image them, get some sort of co-localization map, and then look at the intensity correlation to see if the fluors overlap, and that will use the correlation coefficients we talked about earlier. The first would be Landau's correlation coefficients, and then also the Pearson's correlation coefficients. We can also look at the proximity in terms of distance of the fluorophores, and there's a variety of ways of doing that. And we might need to think about how we apply thresholds that are relevant for our analysis there. We'll use the packages called colloc-R and imager. So colloc-R allows you to compute co-localization metrics like Pearson's coefficient, Landau's coefficients, and other different approaches. And what we'll do is look at things like African swine fever infected vero cells from different microscopy images, which you can see here that there is STED imaging of the major capsid protein P72 with capsid rings in the first image here. And then we can see the capsid rings here in the next image. And then we can merge the images at the end, so you can see that reticular network and the association between the different proteins that are of interest here. And this is from this paper and looking at ASFE and the replication sites. And we'll also look at other images, such as one we saw earlier of the cells of interest and then the virus factories around it. So in terms of measuring the intensity of labelings, our steps then are relatively similar. So we have our fluorescence of interest, and therefore we can think about the structures and stuff that are present. So we want to get our images, identify the regions of interest, pre-process the image using contrast, reducing noise, correcting the background. We'll use R. Alternatives to that are using ImageJ or self-profile to select region of interest. And then we can infer stats from that to compare between different samples. And then we might consider then how sample conditions may have affected that, calibrate these intensities, and interpret them. And you might look at these images that we looked at previously that are here. So that concludes this video.